We now turn to the other enzyme-mediated signaling pathway that leads from GPCRs. The pathway that begins with the activation of the membrane-bound enzyme phospholipase C and leads to the generation of the second messengers, inositol trisphosphate and diacylglycerol. The inositol phospholipid pathway triggers a rise in intracellular Ca2+. Some GPCRs exert their effects via G proteins that activate the membrane-bound enzyme phospholipase C instead of adenylyl cyclase. Several examples are given in Table 16.4. Once activated, phospholipase C propagates the signal by cleaving a lipid molecule that is a component of the plasma membrane. The molecule is an inositol phospholipid, a phospholipid with the sugar inositol attached to its head, that is present in small quantities in the cytosolic half of the membrane lipid bilayer. See Figure 1117. Because of the involvement of the phospholipid, the signaling pathway that begins with the activation of phospholipase C is often referred to as the inositol phospholipid pathway. It operates in almost all eukaryotic cells and can regulate a host of different effector proteins. The pathway works in the following way. When phospholipase C chops the sugar phosphate head off, the inositol phospholipid, it generates two small signaling molecules, inositol 1,4,5-trisphosphate, IP3, and diacylglycerol, DAG. IP3, a water-soluble sugar phosphate, diffuses into the cytosol, while the lipid diacylglycerol remains embedded in the plasma membrane. Both molecules play a crucial part in relaying the signal, and we will consider them in turn. The IP3 released into the cytosol rapidly encounters the endoplasmic reticulum, where it binds to and opens Ca2+ channels that are embedded in the endoplasmic reticulum membrane. Ca2+ stored inside the endoplasmic reticulum rushes out into the cytosol through these open channels. Figure 1625, causing a sharp rise in the cytosolic concentration of free Ca2+, which is normally kept very low. This Ca2+ in turn signals to other proteins, as discussed below. The diacylglycerol that is generated along the with IP3 helps recruit and activate a protein kinase, which translocates from the cytosol to the plasma membrane. This enzyme is called protein kinase C, PKC, because it also needs to bind Ca2+ to become active. See Figure 1625. Once activated, PKC phosphorylates a set of intracellular proteins that varies depending on the cell type. PKC operates on the same principle as pKa, although most of the proteins it phosphorylates are different. A Ca2+ signal triggers many biological processes. Ca2+ has such an important and widespread role as a small intracellular messenger that we must digress to consider its functions more generally. A surge in the cytosolic concentration of free Ca2+ is triggered by many kinds of stimuli, not only those that act through GPCRs. When a sperm fertilizes an egg cell, for example, Ca2+ channels open, and the resulting rise in cytosolic Ca2+ triggers the egg to start development. Figure 1626. For skeletal muscle cells, a signal from a nerve triggers a rise in cytosolic Ca2+ that initiates muscle contraction. And in many secretory cells, including nerve cells, Ca2+ triggers secretion. Ca2+ stimulates all these responses by binding to and influencing the activity of various Ca2+ responsive proteins. The concentration of free Ca2+ in the cytosol of an unstimulated cell is extremely low, 10 to the -7 molar, compared with its concentration in the extracellular fluid and in the endoplasmic reticulum. These differences are maintained by membrane-embedded pumps that actively pump Ca2+ out of the cytosol, either into the endoplasmic reticulum or across the plasma membrane and out of the cell. As a result, a steep electrochemical gradient of Ca2+ exists across both the endoplasmic reticulum membrane and the plasma membrane, discussed in Chapter 12. When a signal transiently opens Ca2+ channels in either of these membranes, Ca2+ rushes into the cytosol down its electrochemical gradient, triggering changes in Ca2+ responsive proteins in the cytosol. The same pumps that normally operate to keep cytosolic Ca2+ concentrations low also help to terminate the Ca2+ signal. The effects of Ca2+ in the cytosol are largely indirect, in that they are mediated through the interaction of Ca2+ with various kinds of Ca2+ responsive proteins. The most widespread and common of these is Calmodulin, which is present in the cytosol of all eukaryotic cells that have been examined, including those of plants, fungi, and protozoa. When Ca2+ binds to Calmodulin, the protein undergoes a conformational change that enables it to wrap around a wide range of target proteins in the cell, altering their activities. Figure 1627. One particularly important class of targets for Calmodulin is the Ca2+ Calmodulin-dependent protein kinases, CAM kinases. When these kinases are activated by binding to Calmodulin complexed with Ca2+, they influence other processes in the cell by phosphorylating selected proteins. In the mammalian brain, for example, a neuron-specific CAM kinase is abundant at synapses, where it is thought to play a part in some forms of learning and memory. This CAM kinase is activated by the pulses of Ca2+ signals that occur during neural activity, and mutant mice that lack the kinase show a marked inability to remember where things are. Intracellular signaling cascades can achieve astonishing speed, sensitivity, and adaptability. The steps in the signaling cascades associated with GPCRs take a long time to describe, but they often take only seconds to execute. Consider how quickly a thrill can make your heart beat faster when adrenaline stimulates the GPCRs in your heart muscle cells, accelerating your heartbeat, or how fast the smell of food can make your mouth water through the GPCRs for odors in your nose and the GPCRs for acetylcholine and salivary cells, which stimulate secretion. Among the fastest of all responses mediated by a GPCR, however, is the response of the eye to bright light. It takes only 20 milliseconds for the most quickly responding photoreceptor cells of the retina, the cone photoreceptors, which are responsible for color vision in bright light, to produce their electrical response to a sudden flash of light. This speed is achieved in spite of the necessity to relay the signal over several steps of an intracellular signaling cascade. But photoreceptors also provide a beautiful illustration of the positive advantages of signaling cascades. In particular, such cascades allow spectacular amplification of the incoming signal and also allow cells to adapt so as to be able to detect signals of widely varying intensity. The quantitative details have been most thoroughly analyzed for the rod photoreceptor cells in the eye, which are responsible for non-color vision in dim light. Figure 1628. In this cell, light is sensed by rhodopsin, a G protein-coupled light receptor. Light-activated rhodopsin activates a G protein called transducin. The activated alpha subunit of transducin then activates an intracellular signaling cascade that causes cation channels to close in the plasma membrane of the photoreceptor cell. This produces a change in the voltage across the cell membrane, which alters neurotransmitter release and ultimately leads to a nerve impulse being sent to the brain. The signal is repeatedly amplified as it is relayed along this intracellular signaling pathway. Figure 1629. When lighting conditions are dim, as on a moonless night, the amplification is enormous. As few as a dozen photons absorbed in the entire retina will cause a perceptible signal to be delivered to the brain. In bright sunlight, when photons flood through each photoreceptor cell at a rate of billions per second, the signaling cascade adapts, stepping down the amplification more than 10,000-fold, so that the photoreceptor cells are not overwhelmed and can still register increases and decreases in the strong light. The adaptation depends on negative feedback. An intense response in the photoreceptor cell generates an intracellular signal, a change in Ca2+ concentration that inhibits the enzymes responsible for signal amplification. Adaptation frequently occurs in signaling pathways that respond to chemical signals. Again, it allows cells to remain sensitive to changes of signal intensity over a wide range of background levels of stimulation. Adaptation, in other words, allows a cell to respond to both messages that are whispered and those that are shouted. Taste and smell also depend on GPCRs. It seems likely that this mechanism of signal reception, invented early in the evolution of the eukaryotes, has its origin in the basic and universal need of cells to sense and respond to their environment. Of course, GPCRs are not the only receptors that activate intracellular signaling cascades. We now turn to another major class of cell surface receptors, enzyme-coupled receptors, which play a key part in controlling cell numbers, cell differentiation, and cell movement in multicellular animals. Enzyme-coupled receptors. Like GPCRs, enzyme-coupled receptors are transmembrane proteins that display their ligand-binding domains on the outer surface of the plasma membrane. Instead of associating with a G protein, however, the cytoplasmic domain of the receptor either acts as an enzyme itself or forms a complex with another protein that acts as an enzyme. Enzyme-coupled receptors, see Figure 1615c, were discovered through the role in responses to extracellular signal proteins, growth factors that regulate the growth, proliferation, differentiation, and survival of cells in animal tissues. See Table 16.1, page 535 for examples. Most of these signal proteins function as local mediators and can act at very low concentrations, about 10 to the negative 9 to 10 to the negative 11 molar. Responses to them are typically slow, on the order of hours, and they require many intracellular transduction steps that eventually lead to changes in gene expression. Enzyme-coupled receptors, however, can also mediate direct, rapid reconfigurations of the cytoskeleton, controlling the way a cell changes its shape and moves. The extracellular signal for these architectural alterations are often not diffusible signal proteins, but proteins attached to the surfaces over which a cell is crawling. Disorders of cell growth, proliferation, differentiation, survival, and migration are fundamental to cancer, and abnormalities in signaling via enzyme-coupled receptors have a major role in the development of this class of diseases. The largest class of enzyme-coupled receptors is made up of those with a cytoplasmic domain that functions as a tyrosine protein kinase. phosphorylating specific tyrosines on selected intracellular proteins. Such receptors are called receptor tyrosine kinases, RTKs, and we will focus on these receptors here. Note that all of the other protein kinases we have discussed so far, including pKa, pKc, and CAM kinases, are serine threonine kinases. Activated RTKs recruit a complex of intracellular signaling proteins. To do its job as a signal transducer, an enzyme-coupled receptor has to switch on the enzyme activity of its intracellular domain, or of an associated enzyme when an external signal molecule binds to its extracellular domain. Unlike the 7-pass GPCRs, enzyme-coupled receptor proteins usually have only one transmembrane segment, which is thought to span the lipid bilayer as a single alpha helix. Because a single alpha helix is poorly suited to transmit a conformational change across the bilayer, enzyme-coupled receptors have a different strategy for transducing the extracellular signal. In many cases, the binding of a signal molecule causes two receptor molecules to come together in the membrane, forming a dimer. Contact between the two adjacent intracellular receptor tails activates their kinase function, with the result that each receptor phosphorylates the other. In the case of RTKs, the phosphorylations occur on specific tyrosines located on the cytosolic tail of the receptors. Tyrosine phosphorylation then triggers the assembly of an elaborate intracellular signaling complex on the receptor tails. The newly phosphorylated tyrosines serve as binding sites for a whole zoo of intracellular signaling proteins, perhaps as many as 10 or 20 different molecules. Figure 1630. Some of these proteins become phosphorylated and activated on binding to the receptor, and they then propagate the signal. Others function solely as adapters, which couple the receptor to other signaling proteins, thereby helping to build the active signaling complex. All of these intracellular signaling proteins possess a specialized interaction domain, capable of recognizing specific phosphorylated tyrosines. Similar protein domains allow intracellular signaling proteins to recognize the phosphorylated lipids that are produced in a membrane in response to certain signals, as we discuss later. While they last, the protein complexes assembled on the cytosolic tails of the RTKs can transmit the signal along several routes simultaneously to many destinations inside the cell, thus activating and coordinating the numerous biochemical changes that are required to trigger a complex response, such as cell proliferation. To help terminate the response, the tyrosine phosphorylations are reversed by protein tyrosine phosphatases, which remove the phosphates that were added to the tyrosines of the RTKs and other signaling proteins in response to the extracellular signal. In some cases, activated RTKs and GPCRs are inactivated in a more brutal way. They are dragged into the interior of the cell by endocytosis and then destroyed by digestion in lysosomes. Different RTKs recruit different collections of intracellular signaling proteins, producing different effects. However, certain components seem to be used by most RTKs. These include, for example, a phospholipase C that functions in the same way as the phospholipase C activated by GPCRs to activate the inositol phospholipid signaling pathway discussed earlier. See Figure 1625. Another intracellular signaling protein that is activated by almost all RTKs is a small GTP binding protein called Roz. Most RTKs activate the monomeric GTPase Roz. As we have seen, activated RTKs recruit many kinds of intracellular signaling proteins and form large signaling complexes. One of the key players in these signaling complexes is Roz, a small GTP binding protein that is bound by a lipid tail to the cytoplasmic phase of the plasma membrane. Virtually all RTKs activate Roz, including platelet-derived growth factor (PDGF) receptors, which mediate cell proliferation in wound healing, and nerve growth factor (NGF) receptors, which prevent certain neurons from dying in the developing nervous system. The Roz protein is a member of a large family of small GTP binding proteins, often called monomeric GTPases, to distinguish them from the trimeric G proteins that we encountered earlier. Roz resembles the alpha subunit of a G protein and functions as a molecular switch in much the same way. It cycles between two distinct conformational states, active when GTP is bound and inactive when GDP is bound. See Figure 1614b. Interaction with an activated signaling protein encourages Roz to exchange its GDP for GTP, thus switching Roz to its activated state. Figure 1631. After a delay, Roz switches itself off again by hydrolyzing its bound GTP to GDP. Movie 16.6. In its active state, Roz promotes the activation of a phosphorylation cascade in which a series of serine/threonine protein kinases phosphorylate and activate one another in sequence, like an intracellular game of dominoes. Figure 1632. This relay system, which carries the signal from the plasma membrane to the nucleus, includes a 3-kinase protein module called the MAP kinase signaling module, in honor of the final kinase in the chain, the mitogen activated protein kinase, or MAP kinase. Mitogens are extracellular signal molecules that stimulate cell proliferation. In this pathway, MAP kinase is phosphorylated and activated by an enzyme called, logically enough, MAP kinase, and this protein is itself switched on by a MAP kinase, which is activated by Roz. At the end of the MAP kinase cascade, MAP kinase phosphorylates various effector proteins, including certain transcription regulators, altering their ability to control gene transcription. This change in the pattern of gene expression may stimulate cell proliferation, promote cell survival, or induce cell differentiation. The precise outcome will depend on which other genes are active in the cell and what other signals the cell receives. How biologists unravel such complex signaling pathways is discussed in How We Know, pages 560 to 561. The importance of Roz has been demonstrated in various ways. If Roz is inhibited by an intracellular injection of Roz inactivating antibodies, for example, a cell may no longer respond to some of the extracellular signals that it would normally respond to. Conversely, if Roz activity is permanently switched on in some cell types, the cells act as if they are being bombarded continuously by proliferation-stimulating extracellular mitogens, discussed in Chapter 18. Before Roz was discovered in normal cells, a mutant form of it was found in human cancer cells. The mutation inactivated the GTPase activity of Roz, so that the protein could not shut itself off, promoting uncontrolled cell proliferation and the development of cancer. About 30% of human cancers contain such activating mutations in Roz genes, and many of the cancers do not produce mutant Roz proteins, have mutations in genes whose products lie in the same signaling pathway as Roz. Many of the genes that encode these intracellular signaling proteins were initially identified in the hunt for cancer-promoting oncogenes, discussed in Chapter 20. RTKs activate PI3 kinase to produce lipid docking sites in the plasma membrane. Many of the extracellular signal proteins that stimulate animal cells to survive, grow, and proliferate act through RTKs. These include signal proteins belonging to the insulin-like growth factor IGF family. One crucial important signaling pathway that RTKs activate to promote cell growth and survival relies on the enzyme phosphoinositide 3 kinase, PI3 kinase, which phosphorylates inositol phospholipids in the plasma membrane. These phosphorylated lipids become docking sites for specific intracellular signaling proteins, which relocate from the cytosol to the plasma membrane, where they can activate one another. One of the most important of these relocated signaling proteins is the serine threonine protein kinase, ACT, which is also called protein kinase B, or PKB, figure 1633. ACT promotes the growth and survival of many cell types, often by inactivating the signaling proteins it phosphorylates. For example, ACT phosphorylates and inactivates a cytosolic protein called BAD. In its active state, BAD encourages the cell to kill itself by indirectly activating a cell suicide program called apoptosis, discussed in Chapter 18. Phosphorylation by ACT thus promotes cell survival by inactivating a protein that otherwise promotes cell death. Figure 1634. In addition to promoting cell survival, the PI3 kinase ACT signaling pathway also stimulates cells to grow in size. It does so by indirectly activating a large serine threonine kinase called TOR. TOR stimulates cells to grow both by enhancing protein synthesis and by inhibiting protein degradation. Figure 1635. The anti-cancer drug rapamycin works by inactivating TOR, reinforcing the importance of the PI3 kinase ACT signaling pathway in regulating cell growth and survival. Some receptors activate a fast track to the nucleus. Not all enzyme-coupled receptors trigger complex signaling cascades that require the cooperation of a sequence of protein kinases to carry a message to the nucleus. Some receptors use a more direct route to control gene expression. A few hormones and many local mediators, called cytokines, bind to receptors that can activate transcription regulators that are held in a latent, inactive state near the plasma membrane. Once turned on, these regulatory proteins, called STATs, for signal transducers and activators of transcription, head straight for the nucleus, where they stimulate the transcription of specific genes. This direct signaling pathway is used, for example, by interferons, which are cytokines produced by infected cells that instruct other cells to produce proteins that make them more resistant to viral infection. Unlike the RTKs that stimulate elaborate signaling cascades, the cytokine and hormone receptors that rely on STATs have no intrinsic enzyme activity. Instead, they associate with cytoplasmic tyrosine kinases called JAKs, which are activated when a cytokine or hormone binds to the receptor. Once activated, the JAKs phosphorylate and activate STATs, which then migrate to the nucleus, where they stimulate the transcription of specific target genes. For example, the hormone prolactin, which stimulates breast cells to make milk, acts by binding to a receptor that is associated with a specific pair of JAKs. These JAKs activate a particular STAT that then turns on the transcription of the genes encoding milk proteins. Figure 1639. Different cytokine and hormone receptors evoke different cell responses by activating different stats. Like any pathway that is turned on by phosphorylation, these signals are shut off by protein phosphatases that remove the phosphate groups from the activated signaling proteins. An even more direct signaling pathway is used by the receptor protein Notch, which controls, among other things, the development of neural cells in Drosophila. See Figure 16.4. In this pathway, the receptor itself acts as a transcription regulator. When activated by the binding of delta, which is attached to a neighboring cell, the Notch receptor is cleaved. This cleavage releases the cytosolic tail of the receptor, which heads to the nucleus, where it helps to activate the appropriate set of Notch-responsive genes. Figure 1640. This signaling pathway utilizes the simplest and most direct way known to transmit a signal from a cell surface receptor to the nucleus. Multicellularity and cell communication evolved independently in plants and animals. Plants and animals have been evolving independently for more than 1 billion years, the last common ancestor between a single-celled eukaryote that most likely lived on its own. Because these kingdoms diverged so long ago when it was still every cell for itself, each has evolved its own molecular solutions to the complex problem of becoming multicellular. Thus, the mechanisms for cell-cell communication in plants and animals evolved separately and might be expected to be quite different. At the same time, however, plants and animals started with a common set of eukaryotic genes, including some used by single-celled organisms to communicate among themselves, and so their signaling systems should show some similarities. Like animals, plants make extensive use of transmembrane cell surface receptors, especially enzyme-coupled receptors. The spindly weed Arabidopsis thaliana, see Figure 133, has hundreds of genes encoding receptor serine-threonine kinases. These are, however, structurally distinct from the receptor serine-threonine kinases found in animal cells, which we do not discuss in this chapter. The plant receptors are thought to play an important part in a large variety of cell signaling processes, including those governing plant growth, development, and disease resistance. In contrast to animal cells, plant cells seem not to use RTKs, steroid hormone-type nuclear receptors, or cyclic AMP, and they seem to use few GPCRs. One of the best studied signaling systems in plants mediates the response of cells to ethylene, a gaseous hormone that regulates a diverse array of developmental processes, including seed germination and fruit ripening. Tomato growers use ethylene to ripen their fruit, even after it has been picked. The ethylene receptors do not belong to any of the classes of receptor proteins that we have discussed so far. They are dimeric transmembrane proteins, and surprisingly, it is the empty receptor that is active. In the absence of ethylene, the empty receptor activates a protein kinase that ultimately shuts off the ethylene-responsive genes in the nucleus. When ethylene is present, the receptor and kinase are inactive, and the ethylene-responsive genes are transcribed. Figure 1641. This strategy, whereby signals act to relieve transcriptional inhibition, is commonly used in plants. Protein kinase networks integrate information to control complex cell behaviors. In this chapter, we have outlined several pathways for conveying a signal from the cell surface to the cell interior. Figure 1642 compares 5 of these pathways. The routes from GPCRs via adenylyl cyclase and via phospholipase C, and the routes from RTKs via phospholipase C, via Roz, and via PI3 kinase. Each pathway differs from the others, yet they use some common components to transmit their signals. Because all these pathways eventually activate protein kinases, it seems that each is capable in principle of regulating practically any process in the cell. In fact, the complexity of cell signaling is much greater than we have described. First, we have not discussed many of the intracellular signaling pathways available to cells, even though many of these are critical for proper development and are deranged in cancer cells. See Figure 2049. Perhaps most importantly, the major signaling pathways we have discussed interact in ways that we have not described. They are connected by interactions of many sorts, but the most extensive links are those mediated by the protein kinases present in each of the pathways. These kinases often phosphorylate and hence regulate components in other signaling pathways, in addition to components in the pathway to which they themselves primarily belong. Thus, a certain amount of crosstalk occurs between the different pathways. See Figure 1642, and indeed between virtually all of the control systems of the cell. To give an idea of the scale of the complexity, genome sequencing studies suggest that about 2% of our about 20,000 protein coding genes code for protein kinases. Moreover, hundreds of distinct types of protein kinases are thought to be present in a single mammalian cell. How can we make sense of this tangled web of interacting signaling pathways, and what is the function of such complexity? A cell receives messages from many sources, and it must integrate this information to generate an appropriate response, to live or die, to divide or differentiate, to change shape, to move, to send out a chemical message of its own, and so on. Movies 16.7, 16.8, and 16.9. Through the crosstalk between signaling pathways, the cell is able to put together multiple bits of information and react to the combination. Thus, some intracellular signaling proteins act as integrating devices, usually by having several potential phosphorylation sites, each of which can be phosphorylated by a different protein kinase. Information received from different sources can converge on such proteins, which then convert the input to a single outgoing signal. Figure 1643, and see Figure 1613. The integrating proteins in turn can deliver a signal to many downstream targets. In this way, the intracellular signaling system may act like a network of nerve cells in the brain, or like a collection of microprocessors in a computer, interpreting complex information and generating complex responses. Our exploration of the pathways that cells use to process signals from their environment has led us to some receptors on the cell surface, to the proteins that form the elaborate control systems that operate deep within the cell's interior. We have examined a large array of signaling networks that allow cells to combine and process inputs from different sources, store information, and respond in an appropriate manner that benefits the organism. But our understanding of these intricate networks is still evolving. We are still discovering new links in the chains, new signaling partners, new connections, and even new pathways. Unraveling these cell signaling pathways in both animals and plants is one of the most active areas of research, and new discoveries are being made every day. Genome sequencing projects continue to provide long lists of components involved in signal transduction in a large variety of organisms. Even when we have identified all the pieces, however, it will remain a major challenge to figure out exactly how they fit together to allow cells to integrate the diverse array of signals in their environment and respond in the appropriate way. In a way, learning how cells think is a problem akin to learning how we, as humans, think. Although we know, for example, how neurotransmitters activate certain neurons and how one neuron communicates with another, we are nowhere near having a fundamental understanding of how all of these components work together to enable us to reason, converse, laugh, love, and attempt to unravel the fundamental nature of the universe and life on Earth. Essential concepts. Cells in multicellular organisms communicate through a large variety of extracellular chemical signals. In animals, hormones are carried in the blood to distant target cells, but most other extracellular signal molecules act over only a short range. Neighboring cells often communicate through direct cell-cell contact. Extracellular signal molecules stimulate a target cell when they bind to and activate receptor proteins. Each receptor protein recognizes a particular signal molecule. Small hydrophobic extracellular signal molecules, such as steroid hormones and nitric oxide, can diffuse directly across the plasma membrane. They activate intracellular proteins, which are usually either transcription regulators or enzymes. Most extracellular signaling molecules cannot pass through the plasma membrane. They bind to cell surface receptor proteins that transduce the extracellular signal into different intracellular signals. There are three main classes of cell surface receptors. 1. Ion channel coupled receptors. 2. G-protein coupled receptors, GPCRs. And 3. Enzyme coupled receptors. GPCRs and enzyme coupled receptors respond to extracellular signals by activating one or more intracellular signaling pathways that alter the behavior of the cell. Turning off signaling pathways is as important as turning them on. Each activated component in a signaling pathway must be subsequently inactivated or removed for the pathway to function again. GPCRs activate a class of trimeric GTP binding proteins called G-proteins. These act as molecular switches, transmitting the signal onward for a short period and then switching themselves off by hydrolyzing their bound GTP to GDP. Some G-proteins directly regulate ion channels in the plasma membrane. Others directly activate or inactivate the enzyme adenylyl cyclase, increasing or decreasing the intracellular concentration of the small messenger molecule cyclic AMP. Still other G-proteins directly activate the enzyme phospholipase C, which generates the small messenger molecules inositol trisphosphate, IP3, and diacylglycerol. IP3 opens Ca2+ channels in the membrane of the endoplasmic reticulum, releasing a flood of free Ca2+ ions into the cytosol. Ca2+ itself acts as a small intracellular messenger, altering the activity of a wide range of Ca2+ responsive proteins, including Calmodulin, which activates various target proteins such as Ca2+ Calmodulin dependent protein kinases, CAM kinases. A rise in cyclic AMP activates protein kinase A, pKa, while Ca2+ and diacylglycerol in combination activate protein kinase C, pKc. pKa, pKc, and CAM kinases phosphorylate selected target proteins on serines and threonines, thereby altering protein activity. Different cell types contain different sets of signaling and effector target proteins and are therefore affected in different ways. Many enzyme-coupled receptors have intracellular protein domains that function as enzymes. Many are receptor tyrosine kinases, RTKs, which phosphorylate themselves and selected intracellular signaling proteins on tyrosines. The phosphotyrosines on RTKs serve as docking sites for various intracellular signaling proteins, usually including the small GTP binding protein Roz. Roz activates a 3-protein MAP kinase signaling module that helps relay the signals from the plasma membrane to the nucleus. Mutations that stimulate cell proliferation by making Roz constantly active are a common feature of many human cancers. Some RTKs stimulate cell growth and cell survival by activating PI3 kinase, which phosphorylates specific inositol phospholipids to produce lipid docking sites in the plasma membrane that allow certain signaling proteins to congregate and activate one another. Some receptors, including Notch and cytokine receptors, activate a direct pathway to the nucleus. Instead of activating signaling cascades, they turn on transcription regulators at the plasma membrane, which then migrate to the nucleus where they activate specific genes. Plants, like animals, use enzyme-coupled cell surface receptors to control their growth and development. Extracellular signals in plants often act by relieving the transcriptional repression of signal-responsive genes. Different intracellular signaling pathways interact, enabling cells to produce an appropriate response to a complex combination of signals. Some combinations of signals enable a cell to survive. Other combinations signal a cell to proliferate, and in the absence of any signals, most animal cells will kill themselves by undergoing apoptosis.